Good evening, guys. Welcome back to my channel. I hope you all are doing well. Today, I'm going to talk about enzyme, its structure, function, and regulation. What is an enzyme? Enzyme are biological catalysts made up of proteins. They speed up biochemical reaction by lowering activation energy, highly specific for substrate and essential for life processes. In easier word, or we can also define it in another word, such as enzymes are biological catalysts that speed up chemical reaction in living organism. Now, structure of enzymes. Enzymes are protein, so some are RNA molecules can act as enzyme or called ribozymes. Their structure determine their specificity and function. Enzymes are globular proteins, active site region where substrate binds. Opoenzymes it is a part of protein. Cofactor is known protein part which has metal ions and coenzymes. Haloenzymes are opoenzymes plus cofactor. Also, we need to know enzymes are protein. Even although some RNA molecule can act as enzymes called ribozymes, their structure determine their specificity and function. There is primary structure of enzyme which is known as linear sequence of amino acid. Then there is secondary and tertiary structure. Secondary is structure is a local folding where A helix and B sheet exist. Then tertiary is the overall 3D structure which forms the active site where substrate binds. Then there is quaternary structure. If it is present, some enzymes consist of multiple subunits. Example hemoglobin. Although not an enzyme, it's a good example of quaternary structure. One of the best example of quaternary structure is hemoglobin because it has multiple subunits. Then there is properties of enzyme. What are the properties of enzyme? An enzyme is a catalyst that increase reaction rate without being consumed. Specificity act on specific substrate, sensitive to pH, temperature, and inhibitors. Reusable can act repeatedly on substrate. Now, what is the function of enzyme? Enzyme catalyzes biochemical reactions, lower activation energy, converts substrate into proteins, essential in digestion, metabolism, DNA replication, and energy production. So, enzyme catalyzes reaction by lowering the activation energy. By substrate binding, substrate binds to the active site, forms enzyme substrate complex known as ES complex. Then there is catalysis. Enzyme facilitates the transformation of substrate into products, does not get consumed or permanently altered in the reaction. Guys, this is a very important part. We need to know that enzyme do not get consumed or permanently altered in the reaction. Product is released and the enzyme is free to catalyze again. So the enzyme never gets finished. It's always present there. It is never completely consumed during the entire reaction process. So no matter how many times the process goes on, the enzyme is not going to get finished. It will be able to rework again and again and again and do the reaction process again and again and again. So it does not get consumed or permanently altered into the reaction. The product is released and the enzyme is free to catalyze again. So no matter how many times we use the enzyme in the reaction, it will surely release the product, but it will be free again to catalyze the reaction. So this part is really important to know. Then there is enzyme regulation. Enzymes are tightly regulated to meet cellular demands and avoid waste or damage. So there is allosteric regulation, there is covalent modification, there is feedback inhibition, then there is inactive precursors. Factors affecting enzyme activity. What are the factors that affect the activity of an enzyme? There's temperature, high heat denatures enzyme, pH, optimal range for each enzyme. Each type of enzyme has its optimum pH, and it works. Every enzyme works best at its optimum pH. Without the optimum pH, without the proper rate of the optimum pH, the enzyme won't be able to process properly. So the each type of enzyme has its own pH optimal range at which it works best. Then there is substrate concentration. Activity increases until saturation. Then there is enzyme concentration, more enzymes, faster reaction. Then there is inhibitors, which is competitive and non-competitive. Then there is regulation of enzymes. What is the regulation of enzymes? As I said, there is allosteric regulation, which where factors bind at sites other than active site. Then there is feedback inhibition. Then there is covalent modification. Then there is gene regulation. What is happening in allosteric regulation? Over here, the molecules binds to a site other than the active site. Then can activate or inhibit the enzyme. Also, in covalent modification, enzymes can be phosphorylated, and enzymes can be phosphorylated, altering their activity, common in signal transduction pathways. Then there is feedback initiation. Uh, we, in feedback inhibition in product of a metabolic pathway inhibit an upstream enzyme, and it prevents the overproduction of the product. So in every regulation of enzyme has its own process, and each of them are important. Also in gene regulation, controlling the gene regulation is for controlling enzyme synthesis. Then what are the applications of enzyme? Where are enzymes really used? Till now we studied that. We got to know that enzymes are biological catalysts that speed up chemical reaction in living organisms, right? Enzymes are protein. So some RNA molecules can act on enzymes called ribozymes. Their structure determines their specificity and function. Also, what is an application of enzyme? Let's see. Industrial fermentation, food processing, detergent, in medical diagnostics, ELISA, drug targets, research, molecular biology tools, DNA polymerase dissection enzyme. So basically over here, enzymes have a wide range of enzymes have wide ranging application across various industries due to their specificity, efficiency, and ability to function under mild condition. Here, so I will tell you one by one. Medical applications, it is used in disease diagnosis. Enzyme assays detect enzyme level as biomarkers. Then there is enzyme replacement therapy, which is known as, I think, ERT, where enzyme is used to treat deficiency disorders, example lactase or lactose intolerance. Then there is bone treatment. Proteolytic enzymes help remove dead tissues, example trypsin. Then enzyme also has a contribution in industrial application, where it is used for detergents, for the for making detergents. Enzyme like proteases, lipase, and amylase break down stains like blood stains, fat, starch. They work at low temperature, and it is also energy saving. Also, it has a contribution in food industry. As I said, lactase break down lactase free milk. Then there is invertase, convert sucrose to glucose, fructose. Then there is pectinase, clarifies fruit juices. Then there is amylases, break down starch into baking and brewing. Then there is textile industries. Also, it has a contribution in textile industries. So let's see what's happening in textile is used in biopolishing, and I think it is used in biopolishing, and there's one more thing in which it is used. I can't remember it right now. Okay, let's move on to the next part. Also, it is used in biopolishing and stone washing genes. It removes fuzz, gives smooth finish. Then there is agriculture application, in where it breaks down phytic acid, improves phosphate absorption, enhances nutrient availability in animal feed. Then there is pesticide degradation. Enzyme can detoxify harmful pesticide residues. So it has contribution in many fields. So enzyme is an important part of life. Also, it has contribution in research in biotechnology, in microbiology, in science. It has contribution in molecular biology. It has contribution in biosensors. It has contribution in synthetic biology. So that's all, guys, for today. I hope these notes help you to prepare well for your exam, because these are really because this PPT is really good for those students who are preparing for exams and who need easier notes regarding enzyme, its application, its process, and its function, and also it's very much easier to memorize from this PPT. So thank you very much. That's all for today. If you like my video, please subscribe my channel and stay tuned for the next upcoming video. Thank you very much, guys. Thank you.